Well, what we hypothesized was that if we could do 3 different relatively simple molecular tests, that we would be able to classify gliomas into meaningful genetic groups, and that those groups would have be meaningful in the sense that they would be people that would act similarly in terms of their response to therapy. They would have similar ages of diagnosis. They would have similar alterations that they might acquire that might be associated with their tumor progression. And what I was most interested in, because I'm a geneticist, is that potentially that a germline predisposition, meaning that the genes we inherit from our parents might be enriched in particular subtypes because presumably the biology would be different. And that turned out to be true. And that we can basically use 3 relatively simple tests to put patients into meaningful groups that's independent of pathology. So in fact, pathology type meaning oligodendroglioma, astrocytoma, mixed oligoastrocytoma, is not very meaningful. So there are oligodendrogliomas that have the molecular features of an astrocytoma. There are astrocytomas that have molecular features of oligodendroglioma. And you cannot you cannot distinguish that through the microscope. But the molecular test is very clear, and when you do that, that can be used to help determine what kind of therapy a patient might need. And what I've been funded by the National Brain Tumor Society to do is try to understand how the germline, IDH, and then the mutations that kind of distinguish a molecular oligodendroglioma from a molecular astrocytoma, how do those parts, I call it the parts list, how do those parts interact to generate a glioma? And that has several reasons to do that. One would be just to understand the biology of how the genome is organized in the brain and how those pieces interact over time to generate the tumor. But also, if we can make what I would call a bona fide glioma that contains the mutations in the right parts, then I think we can develop strategies to find therapeutic approaches, drugs, other small molecules that might specifically target what I call the ensemble of mutations. So where my thinking is a bit different than other scientists is that people think about personalized medicine as targeting one tyrosine kinase, or targeting MYC, or targeting 1 or 2 entities. But in fact, gliomas and almost all cancers are more complicated than that, and they basically have a collection of mutations that drive the tumor, and some of those mutations are early, and some are late. And if one could build a glioma from a set of early mutations, in theory, they must interact in a way that makes the cell susceptible to a particular therapeutic approach. And if you can build the ensemble, then you might be able to inhibit them. And that's what the NBTS is funding, is that kind of pie in the sky idea.